of Veterinary Science and Husbandry from Acharya Nirandeo Agriculture University of Agriculture and Technology, Kumargan, Ayodhya. Uh, Today I am presenting the effect of berberine supplementation on liver and kidney functions of cerebral growth. So first of all, introduce about uh, the importance of this good that is having significant contribution in the economy and food security and poverty elimination of the rural uh, this our Indian people, and India ranked first in terms of this goat milk production and goat population itself also, having a large population like uh, more than 140 million of goat population in the previous as far as 20th life structure is concerned. Very important thing here is the growth rate of this goat population is around 10% from the previous census. And its meat is very preferred and widely consumed meat all over the world. And milk is also used for the medicinal purposes because is the proximity to the milk of the human being, and it's smaller fat globally. So in this, we have taken Sirohi goat to study, and the supplementation, one important supplementation which is an antioxidant, which is quaternary benzoyl isoquinol, that is a flavonoid compound known as berberine. It is obtained from the some important plants of Barbaris, Vulgaris, Equifolium, and Aristata group of the plants, from which we can obtain from root, rhizome, stem, fruit, and every part of the this plant. And if we see about the berberine, its effect from the literature, and found it is a good blood glucose lowering agent, along with its role in the lipid and protein metabolism also, and increasing the antioxidant activity also. So many activities like the anti inhibits oxidative stress inflammation in the tissue, and includes liver, adipose tissue, kidney, and pancreas also. These are the some plants like Berberis, this uh, group of the Indian Barbary and California poppy. These are the plants from which we can get the berberine. And this berberine we can obtain from the commercial feed ingredient with different purity like 70%, 80%, and 90%. And so many other functions like inhibits reactive oxygen species production, and lower the blood cholesterol, LDL cholesterol in the individuals, and also it has reducing effect of the oxidative stress markers like glutathione peroxidase. So with this, we have objective to study the antioxidant status effect, and also effect of berberine on the kidney and liver functions in the this steroid. For this, we have designed a experiment uh, and taken 32 steroid goat with age group of 6 to 12 months, and total duration of the study is 90 days. And all animals, experimental animals, are divided into four groups. First group kept control only fed basal diet. Second group is treatment. First group having supplementation of 2000 milligram per kg diameter berberine, and the third group having 3000, and fourth group having a higher dose like 4000 milligram of the berberine per kg diameter intake. And this blood plasma fluid is uh, 0 day, 15, 30, 45, 60, 75, and 90 day of the experiment. And these are the parameter, these blood metabolites, immunity, kidney functions, liver functions, antioxidant status in the animal. And that directly come to the result in which effect of berberine on the fortnightly body weight gain during the experimental period, in which we have seen uh, the effect of this berberine on the uh, body weight gain. It is found significantly higher body weight gain in the berberine supplemented group as T1, T2, T3 as compared to control group, and we have found almost 17.1% higher body weight gain in the animal as compared to uh, non-supplemented or control group. And we have seen also body condition score because it is also important as far as health of the experimental animal is concerned, in which the body condition score on the basis of different parameters and scale, we have found higher body condition in the treatment group as compared to control group. It is highest in the higher supplemented group T3 followed by T2, T1, and lowest in the control group. Effect of this supplementation of berberine on the antioxidant status of the group during the study period, in which total antioxidant activity it is measured on the basis of the ferric reducing antioxidant property method. And in this total antioxidant capacity was found like a 320 macromole per liter in the T2, uh, T3 group, which is a higher group supplementation group than the medium, lower, and control group. So in this this total antioxidant activity was found significantly higher in the supplemented group, which shows a higher antioxidant status of the group supplemented with the this berberine. And we can see from the figure also the higher level of the antioxidant status in the treatment group as compared to control group. And other we have seen the antioxidant enzyme like SOP catalase superoxide dismutase, which converts superoxide radicals into the less toxic hydrogen peroxide radical. It uh, in this the activity of this superoxide radicals superoxide dismutase is seen in the RBC lysate, in which this berberine supplemented group, the level of activity level of the SOD is found significantly higher, like 15.6 unit per gram of hemoglobin as compared to uh, um, lower doses of the berberine, uh, followed by the lowest level of the SOD activity in the control group. And also, we have seen the goats receiving different doses of berberine at higher activity of the this SOD in the particularly day 55, 60, 75.90 of the experiment period. The second one is the catalase, which convert these hydrogen peroxide radicals into uh, less toxic or non-toxic water. And in this also the activity of this catalase is found significantly higher in the treatment group T3 followed by T2, T1, and lowest in the uh, this catalase. This catalase activity in the control group. And also we have seen the effect of berberine on the liver function, especially LT, EST, and NP. Uh, this is also the marker of liver damages, particularly. So berberine use is useful in the treating fatty liver in the animal. Also, in my experiment, also we have seen that the level of the LT is lower in the treatment group, like 11.09 with respect to 11.96 in T2, in 12.93 in T1, and also higher level is found in the control group. And same result with the AST, it is highest level in the control group. Then uh, T1, T2, and T3. That is, a bargain supplemented group has lower value of the AST. ALP, it is also having a good result in this experiment in which the level of the ALP, like alkaline phosphatase, is found lower in the barbarin supplemented group as compared to control group. In this, all the goat receiving these different doses of barbarin having lower value of these liver enzymes as compared to uh, control group. This shows hepatoprotective function of this berberine supplementation, which is being beneficial in case of the supplementation. This can be seen from the figure also. The variation in the level of ELT, ST, and alkaline phosphatase, it is shown by this figure, and it shows significant higher level in control group as compared to supplemented group. Next is the effect of berberine on the kidney functions like blood urea nitrogen and serum creatinine. In this, significantly overall mean of the blood urea is found significantly lower in the uh, treatment group or supplemented group as compared to control group. And creatinine level, it is also biomarker for the damages in the kidney or muscle metabolism. In this, the level of mean value of the creatinine, it is 1.24 in the T3 group followed by uh, higher level in the control group. It means the supplementation of this berberine is also increases the kidney functions in the experimental animal. These are figures showing the changes in the concentration of the blood urea nitrogen and serum creatinine in the experimental goat during the experimental period. And the effect of this supplementation on the energy metabolism as per is concerned is very important. And in this from literature we have also stated that this berberine is used in the diabetic patient, human being, and also some in laboratory animals. We have seen the metabolism, glucose metabolism also, and found the mean value of the glucose, plasma glucose, 55.88 milligram per deciliter in T3 group, which is significantly lower than the T2, T1, and also the animal receiving not any type of berberine. So from this, we can say berberine has glucose lowering effect in the animal also, and also we have seen non-esterified fatty acid NEFA, which is a goal test for the uh, seeing the negative energy balance in the animal. So from this result, we have seen the significantly lower value of the NEFA in the treatment group, which shows a positive energy balance in the animal. From the figure, we can also see the changes in the plasma glucose and NEFA level in the animal during the study period. And also, we have seen the protein metabolism. For that, total protein, albumin, and globulin level in the plasma we have seen. So total protein level in this experiment, it is found significantly higher in the treatment group than the control group. Actually, this berberine is a flavonoid, and it has structural resemblance with the steroid hormone. So it directly enter into the uh, cell and stimulate the transcription factors or enzymes for the more and more protein synthesis. So supplemental group has also found similar result like the increased protein or functional protein synthesis. The effect of this, it is found in the plasma protein, and the higher level of the protein it is seen. So the same result it found with the albumin also, and significantly higher level of the plasma albumin was seen in the carbon supplemented group as compared to control group. There is no significant variation in the plasma globulin level, but statistically the level of the this plasma globulin is found higher in the treatment group than the control group. This can be also seen with the figures in the given slide. And the effect of this berberine on the cholesterol and triglyceride level, it is very important role in the lipid peroxidation and protecting the cells or cell membrane from the external damages. And in this way also, it has also role in the berberine, the protective role of berberine in the lipid metabolism. In this, we have seen total cholesterol and triglyceride. Total cholesterol level will not found any significant variation, but the level of triglyceride were significantly decreased in the berberine supplemented group as compared to uh, control group. And also, these levels were found significant on different days like 45, 60, and 75, along with the 90 day of the experiment. These also there were those changes in the plasma cholesterol and triglyceride. From this, we can also clearly see in the level of these triglyceride were higher in the control group as compared to berberine supplemented. And also the effect of berberine on the total immunoglobulin level of the survey group during the experimental period. In this, total immunoglobulin level were found significantly higher than it is the mean value is 24.25 milligram per ml. In the last supplemented group, that is T3 followed by T2, T1, and the lowest value of the total immunoglobulin were found in the control group. From figure, it is also seen that the mean value of the total immunoglobulin level were found significantly higher in the treatment group. So from this we can say higher immune status in the animal than the control group. We have also calculated the cost benefit ratio of the supplementing this berberine in the animal. So the market cost of this berberine is almost 5 to 6,000 per kg, and in our experiment it is consuming only 1.8 kilograms. So total cost of feeding almost 10,800, and we have seen the average increase in the weight increase in the term of meat. We have also considered the dressing percentage. So with this we can say extra meat production for 90 days of experimental, it is giving almost 15,485. It is roughly calculated, and in this way net profit is almost 3,485 in the experimental. In this, we have not uh, considered the improved health status which is seen during the experiment, and reduced the treatment cost, labor cost, and also better productive. Yeah, because we have followed up the experiment after after 19 day 90th day B is also, but we have seen the better reproductive also. So in this way, almost 20% of the uh, increase in the benefit, we can say. So with this result, we can conclude the supplementation of berberine reduces oxidative and metabolic stresses, and also improves antioxidant immune status with the hepatoprotective functions in this group. Thank you. This is our experiment photographs. Thank you so much for giving me opportunity. If any query we can take, ma'am.
Hello, sir. You are facing any issues?
Thank you, ma'am.
Thank you so much, sir.